Yes.
Okay, so you can also see the screen. So I'm going to talk a little bit about some of our previous work as background, and also mostly focusing on some ongoing unpublished work. Uh, Most of them are still very rough, but we hope to get some insight from the audience and also the moderators. So our group has been studying lung regeneration and lung repair for a while, and one of the cell type we are very interested in is vascular endothelial cells. And what we think is vascular cells are very heterogeneous, so they are comprised of different populations, not only uh, between different organs and also just within one population, endothelial population at a single cell resolution. It's still very ongoing, but we hope to present some of the unpublished work here. So this picture, this screen is just an example of how heterogeneous lung endothelial cells are. So the green cells are vascular capillary microvascular cells, and in the middle there is a large artery endothelial cell that is marked by red fluorescence. So we hope not only we can study the phenotypic heterogeneity, we can understand how these heterogeneous cells they play different function in terms of regulating lung regeneration and other organ repairs. So why lung regeneration, as shown by a study performed by Dr. Chen Wang 2 years ago, there is actually a lung disease such as just a single COPD, chronic obstructive pulmonary disease in China. There are already almost 100 million patients in China, and there is almost no effective treatments. The only effective treatment now is pretty much just a lung transplant. So this is indeed an unmet clinical need. And what we think is, in addition to the traditional treatment, such as steroid therapy, uh, a promising direction could be a regenerative therapy. So if we can promote the lung regeneration, but how to do this? For all this is pretty much like an ultimate goal for the human beings, how to regenerate an organ. And there is always a hurdle there is there is a scar formation. And what we want is we want to stimulate the organ regeneration and bypass the scar formation, which is fibrosis. So as you all know, right after tissue injury, there is a wound healing response. There is a kind of like a regeneration process, which is growth of epithelial cells. At the same time, there is also a scar formation, which is collagen formation and deposition in the injured organs. And although the scar formation could facilitate later wound healing, but if the scar cannot be removed after the whole process is finished, so there is a fibrosis there. So this persistent fibrosis might hinder the regeneration. So what we think is, if we understand the balance between the regeneration and the fibrosis, we might tip the balance toward regeneration and bypass fibrosis. And one of the regulators we are very interested, as I mentioned from the very beginning, is vascular cells. So we think the vascular cells and probably some blood cells inside the vasculature, they are very active player. They can modulate this process. So by vascular cells, so some of our previous work, we have shown that vascular cells, which is a blood vessel, they're not only a vessel to deliver blood and oxygen, and they also produce a paracrine factor, what we call it is angiocrine factors. They can interact with those cells that are adjacent to the blood vessel, those perivascular cells, epithelial cells, mesenchymal cells. They can always interact with the vascular, especially endothelial cells are lining the vascular lumen. So we think if we understand its interaction better, we probably can modulate the interaction between epithelial cells, mesenchymal cells, and vascular cells in an injured organ, then we can promote the regeneration and get rid of the fibrosis. And what we showed before is we found there is a molecule called MMP14, which is metalloprotease. It can be induced in a lung regeneration model in mouse. So if we remove the left lung lobe of the mouse, of one mouse, the right lobe can regenerate. They pretty much can reform all the alveolar architecture and even the vascular structure in the right lungs. And we think this is probably possibly facultative lung regeneration And what we think is MMP14 molecule expressed by vascular endothelial cells, they play a very active role in terms of regulating adjacent epithelial cell regrowth and also mesenchyme cells without undergoing fibrosis. And this is mediated by a protease dependent release of those paracrine factors, which most are EGF receptor ligands. And like I said, there is very little fibrosis at the end of this process. So this is very hormone coordinated repair process. But we also found if the vascular cells are aberrantly activated in a disease organ, so these abnormally activated endothelial cells, so when they are properly primed, they can produce factors to enhance the regeneration. But in a disease organ, those abnormally activated endothelial cells, they produce a range of fibrogenic, profibrotic factors. So instead of promoting tissue regeneration, so this endothelial cell, they will promote fibrosis. And sometimes they even lead to tumor genesis. So what we think is, so vascular endothelial cells, they play a very diverging role here. And what we want is, we want to understand why sometimes they produce these good factors to promote regeneration, but why sometimes they also produce those bad factors instead of regeneration. Why did they lead to the fibrosis or tumor? And also we want to understand what is the upstream regulatory mechanism. So that leads to what we are doing right now. So we think we are making we are going to two directions here. One is we want to understand this better at the single cell level, and also second, we want to delineate all these mechanisms in a relatively larger animal model, because most of the work we did relies on rodent model, mouse, and sometimes rats. And as you know, rodents are not a perfect model to study the fibrosis, especially lung fibrosis. So what we are doing right now, we are trying to establish a Minipig model using a single cell platform. And why single cell? Why study this at the single cell level? As I said, because we think not only different organ endothelial cells, they are heterogeneous. Even within one organ, lung endothelial cell, they could be very heterogeneous. And we already did a single cell analysis in a regenerating mouse lung. But what we hope right now is we can continue to explore this in a relatively larger animal model, which is a Minipig, as I just mentioned. So we developed a Minipig lung injury model, and by injecting a relatively classical way, injecting those bleomycin, which is a chemotherapeutic drug, through the trachea, and it definitely it led to the lung fibrosis in the Minipig lungs, and we checked the fibrotic response in the Minipig lung through imaging process. This is a high resolution CT image. This shows that after even 3 months, there is a persistent scar formation. The gray area, you can see very clearly, those gray area has been there for a while, which can at least last for 3 to 5 months. So this shows that this lung fibrosis model, it was establishing in this Minipig model. And we also developed a Minipig cryobiopsy platform. So because this is relatively a larger animal, so we could perform a surgical biopsy with this Minipig model, with these Minipigs. And at different time points, we could obtain the lung samples at different time from one same Minipig. So by doing this, we could be able to do we were able to do some multi-omic study, including a single cell analysis. For now, we did a single cell on Sigma, but in the future, we think we could even do a lot of other single cell multi-omic study. And we also collect all the blood and plasma from the same Minipig, and we could do some biomarkers. So then after this, we hope to do some mechanistic study. So because our interest is microenvironmental cells and how these cells regulate the lung repair and lung regeneration. So we isolated all these microenvironmental cells from these from these animals, and we focused on, for example, vascular cells, of course, and also some other blood cells such as B cells, macrophage, neutrophils, and we found there was indeed a lot of change in these microenvironmental cells. And what we want, then we focused on a relatively focused a phenomenon or process, which is epigenetic change, because we think the hypothesis is because most lung fibrosis, they develop, fibrosis develops after chronic injury. So we think not only the genetic change is involved in this process, there should be also some epigenetic change as the chronic injury goes, and there should be accumulation of epigenetic change. And but a relatively superficial study we just did is we focused on the histone modification and also DNA modification. As you all know, because histone acetylation and DNA methylation are probably two most common changes involving epigenetic change. So we collected some samples from the fibrotic lung from the West China Hospital at Chengdu, and we isolated the vascular cells and also lung epithelial cells and checked the epigenetic change from these patient samples. And we found there was indeed some change in histone acetylation in vascular endothelial cells, but not in epithelial cells. So this suggests that at least in these patients, the epigenetic change mostly occurs in the vascular microenvironment cells. So we decided maybe we can, that led to our hypothesis that if we target those epigenetic change, maybe we can normalize the vascular niche, vascular microenvironment, then we can prevent the fibrosis. And indeed, we injected this is kind of like straightforward experiment we just did. We give these diseased animals with HDAC inhibitor and DNMT inhibitors. And we found compared to the disease group, these treated groups, we saw the phenotypic restoration of the fibrosis. Most of the fibrotic responses, they were back to normal. They were gone, and the lung histology looks normal. And we further did this cellular analysis using exactly the same single cell platform. And what we found is there was indeed a lot of changes in these microenvironmental cells after treatment of the HDAC and DNMT inhibitors. And we use bioinformatic analysis and profile all these microenvironmental cells, a single cell resolution. And we found that indeed there is a lot of changes, first between disease group and a control group. Second, also between the disease group and treated group. So the left purple circle is the genetic change between control group and disease group. There is a lot of genetic change in these microenvironmental cells. And the right yellow circle represents the different genetic change between the disease group and also the treated group. And as you can see, the overlapping circle shows that how many genes were restored by this treatment. Which is HDAC inhibitor and DNMT inhibitor. And what we found is compared to all these changes, and vascular endothelial cells seem to show the most restored genes by this treatment. So this data suggests that probably epigenetic change in vascular population, they lead to most change. They play a more prominent role in terms of promoting the fibrosis. So that's what we find so far. And to further identify what is a downstream change and to delineate, to tease out all these the involved mechanism. So we further isolated these Minipig vascular cells using a sorting system. So we are still trying to optimize this system now, but one of the experiments we just did is we isolated the CD31 positive population, which is also CD45 negative and CD31 positive populations from the fibrotic lungs, which should be a bulk vascular endothelial cells that represent both large vessel and capillary vessels. And we did a single cell analysis, especially we compare what is there any change in the treated and the disease group. And we got a relatively surprising outcome is at the very beginning, we expected that there should be a population popping out that promotes the repair after treatment. but what we found is actually there is one single subpopulation that disappeared after treatment. So because this subset disappeared after treatment, and this is associated with improved lung function and lung repair, so we think there might be a bad endothelial subpopulation there that could lead to fibrosis. So then we further analyze this subpopulation with a more detailed bioinformatic analysis, and we find that they could be divided into a couple of subsets. But then further, by looking through all the differentially expressed genes, we find they're mostly related to inflammatory response. And one of a very unique gene, what we find is it's a chemokine called CXCL1. So this is a relatively surprising finding. And as you know, CXCL1 is a chemokine which can recruit a lot of leukocytes and immune cells. And one of its receptors is CXCL2. So what we did, this is pretty much the latest experiment we did. We found if we block its receptor CXCL2 in a mouse model, we find that this also led to a blockage of lung fibrosis. So that's what we got so far. So what we think is if we block this CXCL1 mediated downstream response, it's possible it's probably possible to block the fibrosis. And one of the critical mediator is endothelial CXCL1, which is upregulated by the epigenetic change. So like a oversimplified working model, what we have right now is what we think is, as we showed before, and also in this study, vascular cells, they can form a niche to regulate regeneration of fibrosis. But they could be a good niche or bad niche. What we found before, it mostly depends on what factor they produced. And what we found in this study is even when they produce some bad factors, let's say profibrotic fibrogenic factors, so they could be only mediated by some, not all vascular cells. They probably mediated or be only a subset of endothelial cells. They are in charge of this. So our next step is to further define first, what kind of endothelial cells are these cells, and also second, why they produce these bad factors, not only promoting fibrosis, maybe they're doing something even worse, maybe promoting tumor, for example. So that's our what we think right now. One of the downstream mediator could be these endothelial cells recruit some inflammatory cells and also probably other immune cells here to do all these, to trigger all these downstream events. And for the future direction, of course, we hope we can finish this study and we could figure out and tease out all the molecular details and cellular details. But what we think is the principle and the platform we generated here could be used for body organs. And because a lot of people, they believe there is a tissue-specific endothelial marker like a vascular zip code. But what we think is even within one organ and different pathological and physiological conditions, the vascular zip code could change. What we hope is we could eventually tease all these out, maybe not all of them, but some of them, and at the single cell level, and also use the multi-omic approach to delineate, to define, and eventually to reveal what the functions, what function they do, and why and what is the mechanism that leads to the generation of these factors. That's our future direction. So this work was done is truly a teamwork, and also we got a lot of advice from the Internationl and also domestic advisors and also collaborators. So I probably stop here and would like to get all your insights and suggestions. Thank you.